Hi, Assalamualaikum and a very good morning to everyone. I will be presenting our project entitled Potential Spacer for Electrochemical DNA Sensor, Synthesis and Characterization of p-Nitro Schiff base. Here is the table of contents. DNA detection technology attracts the interest of various fields, such as the clinical, pharmaceutical, forensic research, and many more. The electrochemical DNA sensor offers a good sensitivity, accurate specificity, simplicity, and cheaper diagnosis. To develop a reliable eDNA sensor, it requires a suitable and stable spacer. One of the most common methods to construct a functionalized sensor is by self-assembled monolayer. Self-assembled monolayer is an organized layer of a long chain organic compounds that spontaneously form on a solid surface. They undergo an electrostriation phenomenon when bind with analyte. The most common spacer is alkene-thiol. However, these alkene-thiols face several limitations. Song et al. In 2010 reported that few methods have been developed to attach DNA on the gold surface. However, those methods did not give any good results. In 2016, Lee et al. And Credden et al. Mentioned that alkene-thiols are unstable in long-term storage and at elevated temperature. They are also sensitive to oxidation. Rebecca et al. In 2006 and Ferris et al. In 2009 reported that the increment of alkene-thiol chain may improve their stability, but they will reduce the electron transfer. The choice of anchor strategy determines the stability of the molecular interface. So other than thiol, nitro functional group also have a good affinity to be chemically attached on gold. Thus, the use of nitro as the anchoring group in this study is an important approach to improve the self-assembled monolayer on gold. Since the length and efficiency of electron transfer works vice versa, as mentioned before, therefore we hypothesize that the instability may not only cause from the alkene chain length, but it might also cause from the molecular structure of the spacers itself. The Schiff-based compounds have a broad range of applications, such as in metal complex, anti-cancer, and antibacterial. They were also being applied in electrochemistry and polymer. The Schiff-based aromatic aldehyde were reported to be more stable than the aliphatic due to the effective conjugation system. Therefore, we decided to study a nitro Schiff-based compounds as a spacer. There are three objectives to achieve. First is to synthesize a series of Schiff-based molecules by modifications of their terminal ends. Second is to characterize the synthesized compounds by using FTIR, UV-Vis, NMR, and mass spec. And third is to evaluate the potential of the gold indium tin oxide electrode using the cyclic voltammetry measurement. This study contributes to the development of a new aromatic Schiff-based compounds. They show a remarkable electrostriation phenomenon, and they are also stable and reproducible. This indicates that they are potentially useful as a spacer alternative. Next, the methodology. We first start with the Heck reaction to produce the precursor. Then the product will be used in the Schiff-based reaction to produce the spacer. The final products were characterized by FTIR, UV-Vis, NMR, and mass spec. Then we immobilize the spacers on gold-coated indium tin oxide by self-assembled monolayer. And finally, we analyze the electrochemical properties by cyclic voltammetry. Here is the general structure of our final Schiff-based product, whereby the head group is the nitro functional group, and the terminal is either ethyl, methyl, or methoxy group. From our study, we found out that the FTIR show a significant difference between the precursor and the Schiff-based compounds, especially at the NH stretching and the CO stretching. In UV-Vis, these two bands indicate the CC aromatic of pi to pi star and imine band of N to pi star. From proton NMR, we can clearly see the differences between all compounds, where only the precursor from Heck reaction show a broad peak of NH2 at near 4 ppm, and there is no imine peak above 8 ppm. As for comparison between all Schiff-based compounds, we can see the differences at the upfield region. The results of carbon NMR seems to be in agreement to the proton NMR findings. Other than that, the results of molecular ion obtained from the mass spec analysis were found to be similar to the theoretical values. Therefore, we conclude that all compounds have successfully synthesized. The electrochemical properties of the electrodes before and after being modified were evaluated by the cyclic voltammetry. And here is the voltammogram of bare electrode. After being coated with gold, the current increases. However, we observe a decrease of current after the AuITO electrode being modified by the Schiff-based spacers due to their non-conductive properties. This also means that the monolayer has hindered the electron transfer between the electrolyte and the electrode. Thus, we can also say that the spacer has successfully modified on the AuITO electrode surface. As we can see from the voltammogram, the Schiff-based spacers with methoxy terminal group, which is the black line here, shows the lowest oxidation and reduction peak. This is due to the presence of electron donating group as the substituent. From the peak potential difference, we found out that all these electrodes are quasi-reversible. Meanwhile, the stability of spacers were investigated by carrying out a continuous potential cycling. And we observed that the voltammogram produced are highly identical with R² of 0.9 and RSD of 1.47%. As for reproducibility, we studied by running the cyclic voltammetry on 3 different electrodes that were prepared on the same day under the same condition. The R² was found to be almost 1 with RSD of 2.69%. As a conclusion, we can say that the nitro group has anchored well on the gold surface. However, since the nitro Schiff-based compounds with methoxy terminal shows a very low oxidation and reduction peak, thus we are afraid that we might not be able to distinguish the voltammogram of NO2OMe with the new voltammogram after the immobilization of DNA in our future studies later. Therefore, we conclude that NO2ET and NO2Me are potentially useful as the eDNA spacer. Here are my references. With that, thank you.